This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 92, recorded on November 20th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, how are you, everybody?
I'm well. How have you been?
Fine.
I was in your area twice in the past month.
So I hear, and I didn't get to see you.
It's a shame, right? I should have made an effort.
They keep you busy.
You know, Elio, there are a lot of people out there in the San Diego area. It's pretty crowded.
Yeah.
Is that so?
Yeah, it's just because it must be the weather. I don't know. That laughing voice you hear is from Ann Arbor, Michigan, Michele Swanson.
Hello.
Why are you laughing that I said there's so many people?
People tune in because they're expecting wisdom from us.
That is wisdom. You know, there was a time in the 60s when Jonas Salk went to La Jolla. The governor or the mayor of La Jolla gave him 23 acres on the Pacific to build his institute. And at the time, there was hardly anyone out there. It was an unknown place. Now everyone realizes that the weather's nice and, well, I guess that's it, the weather. What else is out there, right? You got earthquakes.
Hey.
You got a drought. You got locusts. Oh, that's one of our papers today.
Wait with the locusts.
But you know, to finish this story, it turned out that the mayor of La Jolla had promised that parcel of land to UCSD.
Ooh.
So they had a problem, and they decided UCSD had enough land so they could give it to the Salk Institute. Also joining us today from the Medical College of South Carolina, Michael Schmidt.
Hello, everyone.
Charleston, South Carolina.
Where it's cold.
Is that right?
Yeah, it's cold for South Carolina. It's nippy out. We had to break down and put on coats.
But don't you do that in the winter anyway?
Yeah, but this is extremely early. But we think it's going to get nice for next week for Thanksgiving.
All right. Chamber of Commerce weather.
Yeah, well, right now it's 61 degrees, but it feels colder.
It's freezing here. We have winter, as opposed to San Diego, which is 72 and sunny every day. You know, that's boring. Well, it's boring, right, Elio? You have to admit it.
We suffer in silence.
But, Elio, didn't you love living in the Northeast?
I did. I like the change of seasons in Boston.
It's very nice. Okay, today we have some science for you. Hopefully you haven't turned off the podcast because of our little banter. This is just to show you that we're human beings. We're like everybody else. We get distracted. We talk about it. Even Michele gets distracted. The person who you think has the razor focus for any problem, she puts her head to.
I'm right now feeling great that I avoided any questions about the weather here in Ann Arbor.
We know it's like 19 degrees and actively snowing.
Let's talk about the paper.
Well, it's you moved there, Michele. You could have stayed in New York or Boston. That's right. You spent some time in Boston, right?
I did, 10 years. Both my kids were born in Boston.
Do they have Boston accents?
They don't.
Okay. All right. We have a little bit first for you and then a paper. And the little bit has to do with the possible eradication of poliovirus type 3, the Morbidity and Mortality Weekly Report of November 14th, which was just out, has an article entitled Possible Eradication of Wild Poliovirus Type 3. And I just wanted to make a few comments on this. This being the virus, poliovirus, not particularly type 3, that I have worked on for the past 34 years and continue to do so because I have a love affair with poliovirus. It's on my cell phone even. It's everywhere. Now, there are 3 serotypes of poliovirus, types 1, 2, and 3. They all cause paralytic disease. So the vaccines that we have, the Salk inactivated vaccine and the Sabin oral vaccine, they both have the 3 serotypes in them. You have to immunize against all 3. Otherwise, you will still get polio. In fact, in the early 20s and 30s, people were trying to develop polio vaccines without knowing how many serotypes there were. and they failed for obvious reasons. So a crucial finding was that there are 3 and only 3 serotypes. And why there are just 3? Well, in biology, we don't ask why questions, so I don't have to answer that. Anyway, the WHO is on a crusade to eradicate polio. They started in 1988 when there were about 350,000 cases of paralytic disease a year reported, and now we're up to just a few hundred in the current year. So we've done really well. Now, back in 2001, type 2 poliovirus was eradicated. So wild type 2 poliovirus, the last case was in 1999. And if you go 3 years without having a case globally, the WHO says it's eradicated. And that's, of course, somewhat of a random number, but it's based on their experience that if you don't see the virus for that many years, you're not going to see it again.
And humans are the only reservoir of polio, so it's not like we have to stumble into it. We're the only species that has polio.
That's right. Humans are the only reservoir. So this is an eradicable disease because it doesn't exist anywhere else. As far as we know, I will qualify that statement.
Always.
Because it could be under a rock somewhere, right? But probably not. So type 2 is gone. And but you know what? Wild type 2 is gone. There's still type 2 vaccine strains circulating because when you use the Sabin vaccines, which WHO is using in its eradication effort largely, you ingest those vaccines, they replicate in your gut, and then you shed them, and the forms that are shed are no longer vaccines. They're actually virulent again. And those stay in the population for a long period of time, for months or years. And so, interestingly, after you stop... So if you, let's say type 2 was eradicated back in 2001, and WHO stopped using the type 2 component of the Sabin vaccine, but within a year there was an outbreak of type 2 in Nigeria caused by vaccine-derived type 2 poliovirus.
Well, let me ask you, are those people who are, some of them were vaccinated, are they less, is the disease milder?
What do you mean? Well,
when they catch, there's virulent poliovirus around now. Okay,
vaccine-derived, you mean? You're talking about the vaccine-derived polio?
The vaccine-derived polio, yeah.
Oh, it's the same virulence. They get polio. It's just like wild-type polio.
Just like what?
Yeah, it's weird. It's scary. So there were outbreaks of type 2. So WHO's policy now is if we eradicate type 2, we take away the Sabin 2 from the vaccine, but we replace it with inactivated vaccine just to make sure that the circulating vaccine-derived viruses don't cause outbreaks. Now,
but you have to inject it, don't you?
Yeah, unfortunately you have to inject it, which is a big problem, and it's more expensive, and you have to have trained healthcare personnel and so forth. But there's no other way around it because the vaccine-derived viruses will always circulate if you don't do this. So,
in retrospect, was the Sabin vaccine a mistake?
This is really a good question, because right now, this is what we're worried about now, the vaccine itself. Once we get rid of wild polio, the only thing left is going to be the vaccine strains. And so maybe we should have stuck with inactivated vaccine.
But at the same time, you have to think about it from the epidemiologist perspective, because when you vaccinated one individual, they went home and the active vaccine strain came out of them and literally inoculated everyone else in the household. So that probably epidemiologically greatly accelerated the eradication of virulent polio infections in North America and probably led to its eradication in this hemisphere.
Yeah, so this is certainly true, and Israel is a great example of this today. Israel has switched to an activated vaccine. The last 9 birth cohorts in Israel have received IPV. They did a survey last year. They found wild polio in the sewers and in the stool of 30 healthy people. That's because IPV, Salk vaccine, does not immunize your gut. So you don't get polio, but when you're infected with a wild strain, it will replicate in your gut and you will shed it and it spreads. So if we had stuck with just IPV, we'd never get rid of polio. It would probably go gut to gut forever.
And that, from a public health perspective, requires then that you vaccinate everyone so that you don't have these sporadic
that's right.
300 mer
which is,
outbreaks.
as you know, is not it's never possible to immunize everyone, no matter how hard you try. So this is a really interesting conundrum. I suspect that what we did is probably the best thing, to use Sabin and now switch to IPV. So type 3, the last case was in 2012. There were cases in Pakistan and Nigeria, and there hasn't been any wild poliovirus type 3 isolated since then. So another year, and it would probably be declared eradicated. In fact, all of the polio cases this year, the few hundred that have been identified in Pakistan, Afghanistan, Nigeria, and a few other countries, have all been type 1. So assuming that we don't see any more type 3, what this means is now they can use just Sabin OPV type 1 globally, and then follow up.
That which does not present a problem of mutating back to virulence?
Oh, it does, but that is how you get rid of wild virus. It's the proven way.
Uh-huh.
And then you have to follow up with IPV, Salk's IPV, which has all 3 serotypes in it, to prevent vaccine-derived strains from reemerging and causing paralysis. And then when you eradicate type 1, then you can switch entirely to IPV. So I suspect this is going to take another 5 to 10 years. And there's one last thing I want to say, though. So let's say we can get rid of wild-type polioviruses of all 3 serotypes, which I think is possible if we can keep the immunization coverage up high, especially in Pakistan, Nigeria, and Afghanistan, the 3 places where we've never gotten rid of type 1. There still will be circulating Sabin vaccine strains. And if you immunize everyone with IPV, their guts will be susceptible. So I do not know how we're going to get rid of those. I don't see the solution there.
Mm-hmm.
Without coming up with a mucosal vaccine that is not a live virus.
Thank you for saying that. That reminds me to tell you that a study was just done recently where they took kids who had received the normal course of oral polio vaccine, and this is a vaccine that immunizes your gut. They gave them a shot of IPV, and then they challenged them with OPV. The IPV actually boosted their gut immunity.
Huh?
Unexpectedly. So that suggests that if you use IPV, it will immunize the guts of people who previously got OPV, and maybe that'll be the key, and that'll reduce circulation. I'm hoping that'll do it.
Well, let me ask you this question, Vincent. Since polio has been eradicated from this hemisphere for quite some time, has anyone done a survey of our sewage plants to ask the question, is there oral poliovirus vaccine candidates in our sewage, since we're still presently vaccinating our population with the IPV?
This is a very good question. I would look for either vaccine or wild polio, because as people travel, they deposit wild polio. For example, Brazil, during the World Cup, they did surveys of the sewage treatment plants near the airport. Mm-hmm. They found wild polio virus. People are coming in from countries that still have wild polio, right? India, Pakistan, Afghanistan, Nigeria, etc. I asked someone at the CDC if we do this, if we look at the sewers in the U.S., and he said no. And he said the reason we don't is because we don't know what to say if we find it.
Ooh, good. It's a good. Well, it is a conundrum from a public health perspective.
It would freak people out, right?
It would.
Can you see the headlines? Polio is back, right?
And with the recent press on smallpox in the freezer and all sorts of other things that they've been finding in closets, the Congress would go crazy.
So I suspect
I don't understand how come it's not a problem here.
Well, because we have
for the experience elsewhere, it should be a problem here.
We have high immunization coverage, so there's no.
And it's required in order to enroll your child into school.
So I think it's over 95%. So there's no polio as a consequence.
Well, but there are people who don't get immunized, and the virus is probably around.
Probably, I think we've been lucky so far. Yeah, that it hasn't been. There was an introduction of polio into an Amish community in 1979. That's actually the last wild polio outbreak in the U.S. So it's possible.
But I think there's something that works in our favor, which we don't quite understand. Because if you just put on the table what you've said, there should be polio around.
Mm-hmm.
Yeah.
So there's something good happening that we don't know.
How good is our surveillance in this country?
We don't do any surveillance.
So we don't know.
We don't do surveillance unless there's a case
polio
of...
is a reportable disease.
Absolutely.
If a physician sees polio, they better communicate it.
So if you have a child or a person with flaccid paralysis, it gets reported and you have to look for polioviruses. And
Okay.
there's lots of acute flaccid paralysis in the U.S. Every state has a number. In fact, like in California this year, there have been over 100 cases. But they never isolate poliovirus from these kids. But if they did, yeah, it's reportable. You're absolutely right.
So it turns out that biologically, polio ought to be one of the most difficult diseases to eradicate, or the virus at least. But in reality, it seems to work better than expected.
As long as you immunize, it seems to work.
As long as you immunize.
Yeah, the reasons, the countries where we haven't yet succeeded, because immunization has been interrupted. And we have a current outbreak in Syria and Iraq, or a few cases, and that's because immunization coverage dropped. So if we can keep it at higher than 90-95%, it will work eventually. That's right.
Well, I remember when I was a kid, I was vaccinated with both variants of the vaccine. I remember getting the injection for polio, and I remember eating the sugar cube. So I actually got both vaccines out of the Chicago school
So you must
system.
have been born in the 50s, Michael.
I was.
Yeah, there you go.
It shows my age.
I thought you were younger than that.
No, the Chicago was just probably late to the game. I didn't go to school until 1963. So I literally remember getting stuck in the arm in school.
Yep.
And then having the sugar cube some weeks later. So it was the Chicago Health Department that was using both variants. We may have had an outbreak in Chicago and they went crazy doing double duty.
Yeah, I also got both because I was born in 53. I got the Salk vaccine. And then when they introduced Sabin vaccine in 61, 62, they decided to re-immunize everyone. Mm-hmm. So I got that as well. Mm. Anyway, I hope type 3 is eradicated. That would help a lot. It would simplify immunization. But as
it'd be a great success story.
It is. So, but remember, there's still type 3 vaccine viruses circulating, so we have to deal with that.
Well, when I was flying out to the West Coast at the last TWiM, I happened to sit next to an individual who is very much involved in the Rotary clubs of the United States, and he was going out to give a motivational speak about the Rotarians trying to eradicate polio on the globe. And so when he heard I was a microbiologist, he had to tell me his polio, gave me the whole polio lecture. So it was absolutely fascinating hearing it from a Rotarian who was speaking to why it's important to eliminate polio, how this one individual actually met Bill Gates in Africa, and how literally it's because of the polio eradication efforts in Nigeria that the Nigerians, using the command center that they stood up for polio eradication, were able to short-circuit the spread of Ebola.
Yep, it does.
And so I got all of that from my seatmate flying out to give a talk at the American Institute of Oral Biology.
Hey, it's better than getting an infectious disease from your seatmate, right?
It made the coast to coast trip go so quickly, and the person sitting next to us, because I wasn't in the middle, he was at the window and I was on the aisle. I mean, this poor person in the middle was just absolutely fascinated by the discussion. And
you talked across someone, Michael?
Well, no, she was involved in the conversation too, because she was doing what we do on TWiM. She was asking questions of the Rotarian and me, and she said, I have learned so much on this coast to coast trip.
Should have recorded it, Michael.
I should have. I should have got my cell phone out and we could have had a TWiM.
That's right. Could have been an episode. All right, that's polio type 3. Yes, sir.
One more thing is to be said. There, unfortunately, are a significant number of people in this country, and probably elsewhere, who don't believe in vaccination. Mm-hmm. And I'm really sort of worried that this story can be misconstrued. And I hope it's not, because it's very clear that vaccination is what is the success story in the eradication of polio, and nothing else is. But to hear that a vaccine leads to the dissemination of a virus can be misunderstood.
Mm-hmm.
And so I hope that people are nuanced enough to understand what the story really is. Otherwise, I really would worry.
Yeah, well, as you know, there will be some who use that information in a negative way.
Exactly. That's what I'm saying.
Yeah, it always happens. But
Yeah, sure.
at some point in the future, the world will use only Salk vaccine, and
Yeah, we'll be okay.
it'll be okay.
But the take-home message is, again, humans are the only reservoir.
That's right.
And so that's why this strategy is effective.
Yep,
Right.
that's right.
That's a great point, really. That's a great point. Should be kept in mind. It's what made the eradication of smallpox possible, and
Right.
it may not be possible to eradicate Ebola, which probably has a non-human.
An animal reservoir as well.
Yeah, for sure.
Yeah.
That's
But
what I mean.
measles is eradicable because that's another one that only has humans as a host. And in fact, WHO has its sights on measles for the future. But that's harder because there are lots more cases right now, mostly in countries that aren't immunized sufficiently.
And it's probably a function of we don't have two vaccine candidates. We don't have a good mucosal variant as well as a dead vaccine. And I think the polio story is a combination of the fact that we have a live vaccine, which gives you all of the correlates of immunity because you get a real infection, it's temperature sensitive, and you get the correlates of immunity that are necessary to have mimicked a true infection. And then you have the heat killed, which are giving you the right epitopes that the immunologists have defined as being protective. And together, that strategy has really gone to help eradicate a virus off the face of the earth.
Yeah,
Hopefully.
I think we're lucky with polio having two good vaccines. Most vaccines you don't have that, right?
Well, that's a function of the competition that was available in science back in the 50s. You had both Salk and Sabin who were competing.
That's right.
And competition in this particular case really didn't give us one winner, it gave us two.
Yep.
It's also a dreadful disease, so
Oh, yes.
highly motivated to prevent it.
And in fact, our library has on display every so often an iron lung, a true iron lung. And if you've never seen one, go off on the internet and take a look at what the poor victims of polio had to actually live in order to survive this disease.
Hey, Michele, were you there for the 50th anniversary of the vaccine unveiling?
I was not.
You were elsewhere. Oh, yeah. So that was a cool event because the results of the Salk clinical trial were announced at the University of Michigan. Is it Rackham Hall, right?
Yeah.
It's a big deal. And they had a 50th anniversary at which they gave out. So in the first vaccine trial of Salk, they gave the kids who got immunized these badges saying they were called polio pioneers. And Kathy got one at the 50th celebration at Michigan. She gave it to me when I visited.
Oh, that's great.
So I have a polio pioneer badge. Even though
That's a collectible.
It's really cool. It's very, very cool. Thank you,
Well,
Michigan.
My first office here at MUSC was Sabin's old office.
Wow, that's great.
Sabin was here at our institution for a while, so I had Sabin's old office for a while until it was renovated for a mass spectrometer.
Speaking of, this is the last thing we will pursue here. I was just at the Salk, and I visited Clodagh O'Shea, a virologist, and her lab is Renato Dulbecco's old space.
Really?
How about that? This is just great.
Yeah. It's Old Virus Week.
Okay. Let's get on to our paper. Elio, what do you have for us today?
Well, I have something completely different. It's about locusts, their swarms, and what controls whether they swarm or don't swarm. But there is a connection. I'm going to be talking about I'm going to be talking about the extermination or the demise of a biological entity, namely one kind of locust. So if we're going to eliminate viruses, we might as well work on the problems of locusts as well. All right. Locusts. I think everybody knows they were one of the great plagues. They still are. And for a change, let us start with a quote from Exodus 10:15. We don't often use the good book as a source of material, but here it is. It says, "They covered the face of the whole land so that the land was darkened, and they ate all the plants in the land and all the fruits of the trees that hail had left. Not a green thing remained, neither tree nor plant or field throughout all the land of Egypt." Now that is a very accurate description of what happens when swarms of locusts go over the land, and they do still. There are different parts of the world which have this plague beset them. And in fact, I would like to stay a little bit with history one more moment. Can you spare me that?
Of course.
Okay, so the largest recorded locust swarm in history happened in 1875 and covered the states of Colorado, Kansas, Nebraska, and western Missouri. 200,000 square miles. And the swarm that year was, get this, 1,800 miles long and 110 miles wide.
Wow.
And this is not science fiction. This is science
This is not
fact.
science fiction. It sounds like it. 12 trillion locusts. Now, today, these days, the swarms are not that big, but they can be hundreds of miles long still. So it's not uncommon to see something like this repeated. And when they happen, there is really, in those days, there was not a lot they could do. The women threw blankets over their gardens. What did the locusts do? They ate the blankets and then went down and feasted on the plants. They can eat their own weight in plant material in one day. They tried everything. It's really a phenomenal literature. And if you see, in some movies you can see this. The Good Earth, a movie about a book by Pearl Buck, which takes place in China, the locust infestation plays a big role in that. Anyhow, nothing works. You can try fires, you can blast shotguns, you can, this is a good one. You can scour the field with so-called hopper dozers, which happen to be large metal scoops smeared with tar or molasses to grab as many of the locusts as possible. None of that worked. Today, insecticides do work, and they usually spread from an airplane, which is more efficient than any other thing. Now, these farmers of those states affected in 1875 went broke. They had to leave. Many left because there was nothing left to eat. It was just, imagine, nothing green left. So, the odd thing is that the species of locust, which is known as the Rocky Mountain locust, there are plenty of different kinds of locusts, by the way. These locusts, which were present in trillions and trillions, 25, 28 years later, vanished. Okay, they were gone. And the last collection was made 100 years ago in 1902. So there is no safety in numbers. You can go from trillions to zero. Best we know, there are no Rocky Mountain locusts left. And because there were so many, nobody had bothered to collect them much. So there are only a few specimens left in museums.
And they were a pest.
Pardon?
And they were a pest. No one wanted them.
Nobody wanted them, but the entomologists apparently thought, well, I can go out and collect them anytime I want. But they were gone. Now, if people want to study them, they have to go to, I believe it's in Montana, where there's a glacier called the Grasshopper Glacier, and you can find dead locusts there in significant numbers. So why did it disappear? Well, nobody really knows, except that in between swarming, they live in certain restricted parts, and those parts have been essentially plowed under by farming. So it's possible that farming of the site where they live between swarms may have been the reason for the demise. But nobody really knows. I mean, why are they gone? Anyhow, there are plenty of other locusts. So the story is by itself, it's really quite a story. So they come in two kinds. And when I said grasshopper glacier, I said it right. Grasshoppers and locusts are practically the same animal. You call grasshoppers the ones that don't swarm, and you call locusts the ones that swarm. They're not all the same, but there are some which are, the word can be applied to sometimes to both. So anyhow, so what is it that makes them swarm? Well, they aggregate. They're normally solitary beasts, okay? But sometimes they aggregate into these huge clumps, these huge populations. And what does it is a series of stimuli. It's not just a single stimulus, but a series of them. And some are visual. They see each other and they say, I'm going to start liking you. The other has to do with their antenna. When they start touching their antennas, that stimulates them to become aggregate. It's a way of saying, come hither. I like you. But the other thing is chemical stimuli. And these are known as aggregation pheromones, which are found in feces. And they're particularly important. The paper that I'm going to discuss deals with that. And it has a title which is a little bit more complicated than that. It says, Unveiling the Mechanisms by which Microsporidian Parasites Prevent Locust Swarm Behavior, by a series of Chinese plus one American investigator. They are Shi, Guo, Shu, Tan, Miao, Feng, Zhao, Raymond St. Ledger from Maryland, and Wengu Fang. The Chinese people belong to China Agricultural University in Beijing and in College of Life Science of Zhejiang University in Hangzhou. Anyhow, a rather detailed paper published in the Proceedings of the National Academy of Sciences dealing with a phenomenon, namely that the pheromones, the aggregation chemicals found in the feces, are made by bacteria in the midgut. So we have our usual communication between domains, in this case between bacteria and insects, of which there are many examples, and I think there's whole books can be written and will be written about the subject. Anyhow, but in addition, there is one other component, and that is that a protist called a microsporidian, it's actually I'll say something about Microsporidia, prevents the growth of the bacteria. So we have a three-component situation: locust, bacteria, and Microsporidia. Okay, so how does this work? Well, let's first talk about the bacteria. They produce a series of volatile compounds which act to stimulate aggregation. They're different in different locusts, and these ones I studied here are a desert locust, and in it the chemicals are things like nonanal, which is nonaldehyde, a 9-carbon aldehyde, just a hydrocarbon with a CO at the end, hexanal, benzaldehyde, and a few others. Okay, they are. This is the Oriental migratory locust, Locusta migratoria maniliense. That's the full name of it. Anyhow. So, but besides these volatile pheromones, involved are also the neurotransmitters serotonin and dopamine. They're also involved in the transition from solitary to gregarious. So, it's really not easy to study because notice the number of components that are involved in inducing aggregation. As I said, visual, tactile, and chemical. And some of the chemicals, I say, are made by the bacteria, some are made by the host itself. And the serotonin dopamine story is particularly interesting because it looks like serotonin helps initiate the gregarious behavior, and dopamine maintains it. So they work synergistically, which to a neuroscientist ought to be like talking heaven.
Well, one of the things I picked up on when I was reading this, because I always try to compare it to things, is I equated to this as a flying biofilm. And I related it back to quorum sensing, where the quorum molecules, which are relatives of pheromones, go from a planktonic state, which is the non-gregarious form of the locus, to the biofilm state, where you have the gregarious form. And if you just think about, straightforward quorum sensing where a biofilm would form, and then imagine these locusts like a flying biofilm, because that's effectively what these things are, but it's 100 miles long biofilm.
Right.
So, but it's...
I love this. I think this is a great analogy you just made. It helps microbiologists understand what's going on because they're in tune with biofilms. But everybody else should realize that these are classes of phenomena that cut across vast biological entities.
Kingdoms. It cuts across kingdoms.
Domains.
Yeah, domains. That's right.
I'd also like to credit the authors with probably the best first sentence I've read in a science paper ever, which is, the ability of parasites to alter the behavior of their hosts is a widespread phenomenon that fascinates both scientists and non-scientists alike, touching as it does on core philosophical issues such as the existence of free will.
Wonderful. That's a great, you're right.
Isn't that
it's
wonderful?
Really a great piece of writing. I wish they had written the rest of the paper quite that well. It's not an easy paper, but this is a magnificent beginning. And the summary, the abstract also is not bad. It starts, locusts are infamous for their ability to aggregate into gregarious migratory swarms that pose a major threat to food security. All golden words. Very good.
Gregarious, by the way, means fond of company, social.
Uh-huh.
It's a wonderful word that we should use more often.
But I think as Elio is going to take us through the figures, keep this in the back of your mind as you think about the human microbiome and all the complex experiments that our folks are doing. Think about using the strategy that we're going to now discuss, because it really sets the tone of how to design experiments. Because Elio has led up in the prelude, there's a lot of stuff going on in how to design the experiments. These folks are giving a clinic on how to do it.
Yeah, it's very good. It's really a very nice paper. It got quite a bit of attention, so you can find the subject dealt with in a lot of science publications for the public. It's really all over the place, and it deserves it. It's not without some controversy, though. What I read by some people is that by emphasizing the chemicals as the agent of gregarization, of swarming, they touched a raw bone, because some people think that they are not the whole thing. Well, obviously they're not, because, as I say, visual and tactile influences also belong. So it's not easy to dissect it when you think about it. The experiments they do are convincing, but they deal with a laboratory situation. Most of it has to do with putting locusts in a container and a plastic tube open on the top, and then exposing them to volatile compounds and so forth, and observing what passes for gregarization under these conditions, which is how long they stay together, essentially. It is not, in truth, and I think the authors would not mind my saying that, it is not a complete replica of a natural situation. Okay, so keep that in mind. But it's I don't find the paper a problem. I think it's convincing, and often, in pathogenic microbiology, we deal with this all the time. We don't have an animal model system that's suitable, and we do the best we can, right?
Well, even animal models don't necessarily tell you what's happening in people. You have to use a reductionist system like this, right?
Yeah.
So anyhow, it's a good paper. Let's just leave it at that. So anyhow, the story about the bacteria is that they produce volatile compounds, and you can test that. As I say, you put the locusts individually or in groups in a test tube. You expose them to the vapors, to these volatile compounds, and you observe what happens. And sure enough, you can get measurements about their aggregation. You can also get measurements about the behavior of their antennae, which is done by some electrical measurement. And I gotta admit, I didn't quite find it. I looked for it, and I'd have to go back 3 references when I didn't do that. But anyhow, there is a measurement of antenna behavior, which is very highly influenced by the fecal volatiles. So you have aggregation, you have this electrical measurement of the antenna, and you have a few other things that you can measure, which are all telling you the same thing, namely that bacteria produce these compounds, these volatile compounds. The volatile compounds alter the behavior. Now, turns out that if you look at infections of the locust with this microsporidian, that this is all decreased. In other words, the effect is that the bacteria are impeded in their growth. I'll go over that in a minute. And as a consequence, the locusts do not gregorize, do not make swarms as much. And of course, this immediately suggests that the microsporidian are our friends. They cut down on swarming, and therefore they could be used for biological control. And there is talk about that. This is actually being studied and being done. I don't know how far it's going.
Elio, this Microsporidia is a known parasite of locusts out in the wild, right?
Yes, it is. Yes, it is. And I better say something about Microsporidia, shouldn't I?
Sure.
Okay, they are a group of organisms that are small, protist-like. However, it's been very controversial where they belong to. Now they're said to be something like fungi. They are closer to the fungi phylogenetically than anything else, and so having traveled all over the map, that's where they end up. But they're not typically fungi, and they are very, very important parasites of plants and insects and some other animals. In people, you don't find many cases unless you're dealing with very immune-compromised people. There is a microsporidian called Encephalitozoon cuniculi. Michael, you've probably run into that, haven't you? Or have you?
No, this is new. This was new to me. And as I went and looked at these Microsporidia, I was taken aback by how small the ones are that infect mammals. They're down in the bacterial size. They're 1 to 4 microns.
They are not much more than 1 micron across, and they are as small as it gets. They are really the smallest of the eukaryotes. They are eukaryotes, however, you don't find mitochondria in them. And this, of course, opened the debate in the past about whether they are something different. They'd never had mitochondria, but in keeping with Giardia and other organisms that are amitochondriate, that is, they have no mitochondria, the idea is that they had them and they got reduced into something called mitosomes, which are not non-DNA containing organelles. Anyhow, Microsporidia are interesting, and they are very aggressive parasites. So we should probably spend some time bringing a microsporidiologist on board and have us tell us a little bit more about them. Anyhow, so how does this work? So the Microsporidia find themselves in various parts of the animal. They live in the midgut, but they can be found elsewhere. They spread to other parts of the parasite, and eventually they hurt the animal. That is, eventually the Microsporidium causes impaired development, mobility, and decreases reproduction. However, for a long time they don't do that, and they are passed from insect to insect via cannibalism of dead insects. As I said, locusts will eat everything, including their own. Or they can also be passed by fecal contamination and even by transovarial transfer from the infected females to their offsprings. In other words, they are very adept at vertical transmission. So, how do they work? As I said, they impede the growth of the bacteria, leading to a shift back to solitary behavior. Okay, so by studying these things in these chambers containing the volatile chemicals, they can study aggregation, again, to repeat themselves, aggregation, antennal response, and so forth. So they found that parasites a parasite-infected locust makes smaller amounts of the volatile pheromones, the aggregation pheromones. How do they do that? How does this happen? Well, the parasites reduce the pH in the hindgut from 6.3 to 5, which is significant. And this alone inhibits the growth of the bacteria in the gut. So you have fewer bacteria, and in fact, by direct count, a lot of the bugs in the locust are cultivable. And you reduce their viable count by about 20%, which seems to be significant. And the amount of neurotransmitters which is made as a result of the infection with the Microsporidium is about half. You cut the neurotransmitters down to half. So that's, it's how it works. You have the bacteria, which make the pheromones. You have the parasite, which inhibits the bacteria. There is more to it. There's also a reactive oxygen species involved, which are toxic to the bacteria.
If I was on this student's committee, I would have asked them, what would have happened if you gave the locust a proton pump inhibitor? Would that counteract the effect of the Microsporidia to really go after the pH question while still having the infection there? But I was reading this while the TV was running in the background, and in full truth in advertising, I heard a proton pump inhibitor ad.
Yeah.
But that's how you make these connections with the science. But the pH, it's what's so fascinating about this is a subtle change in an order of magnitude change in the acid concentration causes this phenomenal behavioral change that could literally save our food supply.
Yeah, that's right. Sure. I wonder if a proton pump inhibitor wouldn't kill the locusts.
But that could be true, too.
Which is another desired effect. But,
yeah.
Imagine if you're going to hit a eukaryote, it's going to be tricky. But you're right. This would be an interesting thing to look into. So this is pretty much the story. They also did a huge amount of work. This is a paper which has a supplemental material component, which is it's formidable.
34 pages.
Right. You can spend a lot of time. So there's a lot of work here. And in reality, it's important and interesting, and I'm glad they did it, but it does not alter the general picture. So what they did is they looked at the transcriptome of microsporidial infected locusts, compared them with non-parasite-carrying locusts, and they found big changes. Of the 1,600 genes that they looked at, half were upregulated, half were downregulated. And the top 3 regulated ones were genes involved in the metabolic process of organic substances, the binding of heterocyclic components, and certain hydrolase activities. Now, 5 of the genes involved in peroxidase activity were downregulated. And given that the role of peroxidase is to detoxify reactive oxygen species, the decrease could lead to an elevated level of reactive oxygen species. And this in time, in fact, can lead to the increased toxicity to both the bacteria and the parasites. Okay, so it's complicated and it's interesting.
Elio, can I ask you something?
Sure.
I understand why the Microsporidia might displace the bacteria, but is the behavior modification of any use to the Microsporidia?
That's a good question.
The selection pressure question.
It is not, because gregarization would be a way to spread the Microsporidia. Mm-hmm. So yes, this is looks like a not to be to the advantage of the Microsporidia. However,
and actually, we can. I'm sorry to interrupt. Go ahead, Elio.
Oh no, you go ahead.
To take it one step further, this isn't a case of the microbe kind of zombie behavior, changing the behavior of the host to its own good. It's rather a host response to the parasite infection. So if the hosts now leave the swarm and go off by themselves, they're less apt to spread that parasite to their swarm mates. And so the authors close by speculating that this is actually part of a host defense pathway.
Right.
Maybe.
To prevent the spread of infectious agents.
Yeah, it's not good for the parasite, but it's good for the host. Sure. On the other hand, you wonder, do the locusts benefit from swarming? Obviously, they might. So this is not easy. The ecology and evolution of what goes on here is really quite complicated.
I would think the individual locust might suffer, but the swarm would benefit from isolating the sick one.
That's right.
And it's interesting to me that the behavior change is co-regulated with this increase in reactive oxygen species, like 2 different host defense pathways. The behavioral one and the reactive oxygen species are both induced. Right.
Well, this is about it. So I'd like to end with repeating what Michele brought up, what they write, namely that the ability of parasites to alter behavior is widespread, and it touches on core philosophical issues like the existence of free will. Do we have free will, or are we being told what to do? Let me tell you what
my wife tells me what to do.
When the Yiddish writer Bashevis Singer was asked once, "Do you believe in free will?" he said, "Do I have a choice?"
Wonderful.
So that's about the story.
But Elio, we did a paper a couple of months ago on how your gut microbiome might control your craving for chocolate, and you didn't like that, right?
This is exactly that kind of stuff. It's that kind of stuff. Now, I would like readers to really go and Google locusts, and there are movies plenty, YouTube about showing locust swarms. It's a fascinating story, and I think we all would enjoy it.
Okay, yeah. Cool. Thank you, Elio. That's a very, very nice paper. We have a couple of emails that we'd like to answer. The first is from Dave, who writes, Laureen Mascola's distinctive voice and laugh has won my heart this month. I found that when TWiM came up for the 3rd time in my playlist the last couple of weeks, I kept listening just for a fix. She should be a star. I'm glad you had a good reason for featuring her and that she had a good reason for being with you. Great discussion, too. Dave from Fresno. So that was a great episode 2 episodes ago, Laureen Mascola and one other guest from Cepheid, I can't pronounce it. Michael, Cepheid. Really cool stuff. Laureen is the head of ID at the Los Angeles County Department of Public Health, and she talked all about preparedness. And David from Cephi had talked about this amazing PCR assay that they've developed. So go check that out. Ron
it's
writes,
a really cool instrument.
Oh,
I have
amazing.
two in my lab that when we were doing the copper study, the Army was gracious enough to get us those instruments, and it's all microfluidics, and it does an individual reaction, and you have your result really quickly. It's really a cool system.
Go listen to that episode if you haven't, because it's really amazing. It's just an hour. We were under time constraint. It's quick and focused, and it's amazing stuff for both speakers. Ron writes, sorry if this question is so elementary it is boring, but are the terms autotroph and prototroph synonyms? I read somewhere that early on the two terms were used synonymously. Unfortunately, that reading did not go on to describe any differences in the two terms as they are used today. I understand that prototrophs can make all their necessary organic molecules themselves and therefore do not need organic molecules in their environment. Do autotrophs only make their own food? Are there autotrophs that must take certain other organic molecules in from the environment? For example, amino acids or vitamins? Thank you.
When I was a young graduate student, I was always taught by the photosynthetic folks that an autotroph is operationally defined as a microbe that uses CO2 as its sole source of carbon. And that was their absolute, while a prototroph could use various forms of carbon, whether it be CO2, carbon monoxide, or even other reduced organics, and then use that to effectively make the cell. So they could be synonyms, but operationally, I was always taught that the autotroph would only use CO2 as its source of carbon, and its source of carbon would mean how it made more cell. I think where oftentimes people get confused is the term autotroph and auxotroph, whereas the auxotroph is the antonym of prototroph, where it's unable to make a particular amino acid or vitamin or something along those lines.
Is that good, Michele?
It is.
Elio?
Yeah.
Beautiful. Thank you. Ron is at Rutgers University. Next letter is from Juan, who writes, Hi, Twimsters. I'd like to make a follow-up comment about what Dr. Swanson's comment that it doesn't matter what kind of bacteria are in any environment, but solely what sorts of metabolic pathways are there. I've heard this argument before, but I consider it not to be completely true because if that was the case, then we would not have such diversity. I believe that this diversity, regardless of the repetition of metabolic pathways, is because each bacterium has a specific program which will be executed on specific conditions, diet, health, etc., depending on the bacterium. So we want to know what bacteria are in there, which pathways do they have, and under which conditions they are invoked.
I agree with you, Juan. It is valuable to know who is there, but once we know who is there, it will be important to learn what they're doing. In part because of the diversity you point out, many different microbes could provide the same function. And so without knowing the key metabolic contributions, it will be difficult to pull patterns out of the complex microbial community data that we're currently collecting in experiments. Is that clear to you others? Do you have more to add?
I think you need to know both, and I think your comment was meant to emphasize that you can't just know the bacteria; you have to know the pathways that are involved, right?
But certainly knowing the bacteria is a great place to start.
Yeah.
And the microbial ecologist will probably send us a companion letter arguing about the importance of the niche, because as you have a mixed culture, one microbial species may be doing something that is then preparing the local environment for the next microbe. And we see that with archaea feeding the bacteria and vice versa, and consequently the niche or the local environment in which these individual genus or species or strains may actually be located in is almost as important proximity in the sense as what metabolic pathways are there, what microbes are there. So we can't ignore it because the microbial ecologists will send us letters.
So studying each member in pure culture will be not very informative, is another way of emphasizing your point, right, Michael?
Yes. And in fact, Hungate was the most famous for that. He would never use pure cultures. He would, send his students out to the mud each time and have them get a fresh inoculum from the mud to start their experiments rather than working with pure cultures.
It's Pasteur versus Koch.
Yes.
It goes back 150 years.
Yeah.
All right, our last letter is from Judy, who writes, "Hi all, I have several friends who take capsules of probiotic bacteria, Lactobacillus acidophilus, and the star, apparently, bifido. I've asked them, and I ask them to ask their doctors and nutritionists, but no one answers my two major questions about these probiotics. One, what do these bacteria do in your gut? Two, how do they get to the gut if you are taking them in gelatin capsules? Don't they die in the stomach acids?" All right, who's going to tackle this?
Well, first, the Lactobacillus acidophilus, as its name would imply, loves acid. So it can actually pass through the stomach lining. The acid will begin to digest the gelatin capsules, and some folks that are advocating probiotics are actually, they're designing these capsules so they make it through your stomach and only begin to dissolve, or the gelatin is degraded in the small intestine, so the microbe is liberated. The other thing is you often in these capsules have billions of the particular species of microbe, and so you may be able to kill a fraction of them, and if you kill 99.9 and you have a billion, you still have well over 1 million microbes to do their deed. At issue really over acidophilus and bifidobacter is a pretty intolerant anaerobe. It doesn't like too much oxygen, and so if you look at it cross-eyed with too much oxidative stress, it will die off. And so you really have to look at the shelf life of these bacteria. And what they do is they're effectively preparing, and you can go and look at some of the microbiome literature looking at the short-chain fatty acids that these microbes actually produce that are actually beneficial to both the enterocytes in our gut as well as the other beneficial microbes in our gut. And they actually, it goes back to that discussion from the last letter about preparing the local niche, and they're major contributors of preparing the gut for the right circumstance. Others?
I'd say a big question in the field now, Judy, is how many of these bacteria would you have to take, how frequently, and under what conditions for them to actually colonize and become members of your community. It's a real challenge once an established community is developed in your gut to manipulate that by adding new members. So your questions are great.
So has anyone done a study where they look to see whether taking these probiotics leads to changes in colonization?
Oh, sure, they studied it. A book just appeared with the title Probiotics and Prebiotics. I haven't gotten it, but there's a whole book about this. It's a huge amount of information. And as a bystander, it's not my field at all, but as a bystander, I have the impression that the last word is not in yet.
Yeah.
I was actually going to suggest a paper for the next TWiM that I recently stumbled across. It's a paper from January 2013 that's entitled Intestinal Microbiota Containing Bartonella Species Cures. And the editors of I&I left the word cures, vancomycin-resistant Enterococcus faecium colonization in humans. And so that this is along the lines of probiotics. The Bartonella was a species I hadn't been acquainted with, and as I was scanning old volumes of journals that accumulated on my desk, I said I hadn't seen that paper, and so I was going to commend it for our next TWiM along with a few others.
Who would be a person to get on that would be an expert in the use of probiotics?
Probably Eric Paymer from Sloan Kettering, who's the senior author on this particular paper. He's been doing a lot of work with encapsulating, doing stool transplants at Sloan Kettering and other things. So Paymer from Sloan Kettering, who's in the infectious disease service in the Department of Medicine, may be just the person who could comment on that.
And I'll add that Gary Huffnagel here at Michigan has written a book for the public on probiotics. So,
Okay.
Huffnagel may be good too.
Would you think Gary would join us, Michele?
oh, sure.
Yeah, you could influence him, right? Yeah, I think we should get someone who's an expert to talk about this, because I'd like to know the down as well. Because as Elio said, I thought the word was not in yet, but let's see.
And the other part that's tricky is that the probiotic industry is not well regulated, and so it's
Right.
difficult to compare
Right.
across when you've got variability in the population,
Yep.
what their rest of their diet is, and then what they're actually consuming. It's difficult to tease out the effects.
Yeah. Okay. Judy is a loyal TWi ×3 listener from the start. I used to be a high school science teacher, but I have retired and now spend time gardening and volunteering with little kids to teach science. It's a blast, but they ask hard questions. She's in San Diego, where it's 71 degrees, light breeze, sunny, 63% humidity. Yes, another day in paradise. I know you don't do weather on TWiM, but I wanted to brag. Thanks for all you do. We used to get lots of letters from Judy on TWiV, and she's retired. She used to use TWiV for professional development to come up with stuff to teach her kids.
Neat.
I'm sorry that you have retired, Judy, but keep on
I'm
listening.
happy for you, Judy. Enjoy. More time for TWiM and TWiV and TWIP.
Keep on writing. Thanks a lot. This episode of TWiM will be found at iTunes and also at microworld.org/TWiM. And you can send us your questions and comments. We do love to get them. Send them to twim@twiv.tv. And Michele Swanson is at the University of Michigan. Thank you, Michele, for joining us.
Thank you.
Elio Schaechter is at the blog Small Things Considered. Thank you, Elio.
My pleasure. I sure enjoyed reading about
Locusts, huh? Yeah, it's great. And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
Good to have you back. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.